Hello guys, today we are going to prepare Tris buffer, which is a pH stabilizer for the environments for proteins and DNA. We are going to prepare for 100 ml amount which we are take and how we are preparing now we can see. And the pH is from 7.4 to 8, which is mainly used for DNAs and proteins, SGS page, native page, etc. And the apparatus required are Tris buffer, which we are going to prepare, and measuring flask, conical flask, distilled water, and wash bottle. We need butter paper for weighing the tris. Okay, we are going to take the tris for 100 ml. So we are going to take 12.11 grams to make 100 ml tris buffer solution. So to maintain the pH, we are going to use pH meter. I will show in the video when we are going to measure the pH. For weighing the tris buffer or anything, we have to take the butter paper, which will be non-sticky, and we have to make it as paperboard with for not filling out, so it can be easy. So now we are going to keep the butter paper. So the some way will be shown here. So we need to make it 0 and start measuring the tris buffer. Now we got our 12.11 grams. So now we transfer into our conical flask and we will mix it. So now we have to take a 80 ml of water using measuring flask and transfer it into the conical flask. Why 80 ml not 100 ml means after after mixing it thoroughly, we can make it up to 100 ml. So no problem in it. Slowly we have to transfer our tris into the conical flask without filling it. So this is why the butter paper as well as the paperboard formation is helpful for us. So we have to mix it thoroughly until all the solid crystal particles dissolve in it. So now we have to check the pH of the buffer which we have prepared, which is very much important for maintaining the environment for DNA as well as proteins. So we can go there. The pH meter setup. This is the electrode which we going to measure the pH. We need to transfer into the distilled water now in order to check the pH of the distilled water. It's going 7.8 something, right? After getting it, because this is a solution which is need to dip into the electrode. So then we are going to transfer into our tris buffer. Now we can check the pH of our tris buffer. It is going into way long negative like this basic pH. So we need to maintain it in up between 8 to 9. So we are going to add one normal of HCl in dropwise manner to adjust the pH to 8 to 9. So we can go there and add the HCl. As of now, pH is from to 11. We need pH from 7.8 to 8.8. So in order to make it, we are going to add dropwise addition of one normal HCl. It is very concentrated, so we have to handle very carefully. So by adding that, simultaneously we have to check the pH too. So I am adding drop by drop. Adding one drop and we have to mix it. You can see the change in the pH, right? One drop and we have to mix it. The same one drop and we have to mix it. One drop, mix it. You can see the change in pH drastically while mixing the one normal HCl. You should not pour the HCl in your hand, you know what will be the cause of it, right? So handle it carefully. And while pouring the HCl, you should not pour it in the electrode. You should pour only in your solution which you prepare. Still, we need somewhat less in pH for our desired nature of the tris buffer. So we can see now our pH is 8.4, which will be an ideal one. And now we have made our tris buffer. It's not thoroughly 100 ml. So we have to make it as 100 ml, right? So we can add distilled water in order to make it 100 ml. So we can see our final solution in conical flask. 100 ml of our solution. We know that we have taken 80 ml of distilled water. So there will be slight change from 80 to 90, you can see, because we had HCl and our salts also take some volume. So now we have to make 100 ml. So in order to make it 100 ml, we are using distilled water. Okay, now we have got our 100 ml. Now we are going to transfer into our conical flask. We have prepared our 100 ml tris buffer solution. Ideally, we will make our tris buffer solution to 1 liter. So, in order to take 1 liter, we need 121.2 grams of tris buffer. We have to store the tris buffer in freezers and other things.